Principles of Inheritance and Variation, Session 21. This module, we are going to learn about linkage and crossing over. Thomas Hunt Morgan was an American evolutionary biologist. He was a geneticist and embryologist who made many discoveries to prove how chromosomes play a very vital role in heredity. So, along with his colleagues, Thomas Hunt Morgan provided experimental verification on chromosomal theory of inheritance. And his experimental material was the fruit flies, the tiny fruit flies, and its scientific name is Drosophila melanogaster. And this flies is commonly observed or it is seen in and around our houses flying near the fruits. So his experimental material was the tiny fruit flies. He provided convincing evidence that the Mendelian factors, the heritable Mendelian factors were located on the chromosomes. And why he selected the fruit flies was that they breed at a very fast rate. They provide a large number of progeny flies within a single mating. A new generation can be bred every 2 weeks. That means they have a very short lifespan. They complete their lifespan within 2 weeks. And they have only 4 pairs of chromosomes. They have only 4 pairs of chromosomes, and the male and female can be easily distinguishable by the external morphology or the external appearance. So let us once again recollect why Thomas Hunt Morgan selected the experimental material as the fruit flies. They can be grown on simple synthetic medium at a very fast rate in the laboratory using synthetic medium. They complete their life cycle within 2 weeks. A single mating can produce a large number of progeny flies, and the male and female can be easily distinguished from the external morphology. That means they exhibit sexual dimorphism. And many of the heritable variation could be easily observed under a low power microscope. So these are the reasons why Morgan selected the fruit flies. Morgan conducted many dihybrid crosses in Drosophila in order to study the genes that were sex-linked, or the genes which were located on the sex chromosomes are called as the sex-linked genes. So he conducted many dihybrid crosses in order to study the inheritance of sex-linked genes. And he also the crosses conducted by Thomas Hunt Morgan was similar to those conducted by Mendel. And he conducted a hybridization experiment between a yellow-bodied white-eyed female and a brown-bodied red-eyed male. So the two characters he selected was the body color and the eye color. The body color and the eye color were the two characters he studied, and such a cross is called as a dihybrid cross. But he found that the genes did not segregate independently. As conducted by Mendel, he did not obtain a 9:3:3:1 ratio. There was a significant deviation from the 9:3:3:1 ratio in the F2 hybrids. So he wanted to understand what is the reason for this deviation. So he found that the genes for the eye color and the body color, they are situated on the same chromosome. They are situated on the same chromosome, and the chromosome is the X chromosome. As we know, the X chromosome is a sex chromosome. So the genes were located on the X chromosome, that is a sex chromosome, and that is why there was a significant deviation from the 9:3:3:1 ratio. So the genes were present on the same chromosome. They have a tendency to get inherited together, and such genes are called as linked genes. And this phenomenon is called as linkage. So linkage is the property shown by two genes which are located on the same chromosome to get inherited together is called as linkage, and such genes are called as linked genes. Or it can be defined genetically as a failure of two genes to assort independently. They do not separate from each other. They do not get assorted independently. So the failure of the two genes to assort independently is called as linkage. So linkage occurs when the two genes are located close to each other on the same chromosome, and when the two genes which are located on the same chromosomes are called as the synthetic genes. So two genes which are located on the same chromosome are called as synthetic genes, and two genes when they are close to each other, they show on the same chromosome, they have the property called as linkage. So linked genes are surely synthetic. So that means linked genes means two genes are located close to each other and they are located on the same chromosome, so they are called as synthetic genes. But synthetic genes, so genes which are located on the same chromosome, they are not always linked. That means if there is greater distance between the two genes, they are synthetic but they are not linked genes. Greater the distance between the genes, they have the tendency to get assorted independently. So they show less linkage. Closer the genes, they have greater linkage. Farther away the genes, they have lesser linkage. So these genes were located on the same chromosome, that is, they were located on the X chromosome. So nearer the synthetic genes, greater is the chance for linkage and lesser the chance for crossing over and recombination. As we know, crossing over. What is crossing over? We had studied crossing over in meiosis, which takes place at the time of gamete formation during the packaging stage, during prophase packaging stage, in which the homologous chromosome, they get synapsed, they form a connection called as synapses, and such synapses occurs between the non-sister chromatids, the non-sister chromatids of a homologous chromosome. And at the stage of synapses, at the point of synapses, they start to exchange, they start to break off from each other at the point of synapses, and there is crossing over taking place. There is crossing over taking place between the non-sister chromatids of a homologous pair. So smaller the distance or very less distance between the genes located on the same chromosome, they have greater chance for linkage and lesser chance for crossing over and recombination. And what is recombination? The rearrangement of genetic material at the time of crossing over is called as recombination. Between the non-sister chromatids of a homologous pair is called as recombination.